Well, we're very pleased to have Dr. Omar Harb here to talk about malaria in the classroom, and he is the Education and Scientific Outreach Manager for the Eukaryotic Pathogen Database. Dr. Harb has been running workshops for scientists. He serves as an advisor for developing online research tools, and he ensures that, in general, big data are available and readily usable for the scientific community. Dr. Harb's training is in microbiology and molecular biology, and he has special expertise in toxoplasma and Plasmodium parasites. And so let's just turn this over to Dr. Harb.
Okay, thanks a lot, Kristen. It's a pleasure to be here, and I'll be happy to talk to you about Plasmodium, the causative agent of malaria, and take you through the life cycle of the disease, and then ultimately I'd like to talk to you about genomics of Plasmodium. There is a really good resource out there for genomics and Plasmodium that allows you to run your own searches and queries. We have an exercise that takes you through one such query, which you can make as simple or as sophisticated as you want. But the goal, so the lecture is going to be fairly straightforward and simplified maybe, but the goal is really to point out some key talking points when you talk to students in the classroom about where you can potentially expand into a full lecture just on that one topic or one item that's presented in the slide. So just by way of background, so malaria, people in this country, at least in the United States, we're very comfortable to say, well, there's no malaria here, why should we care? And so there are a few things that I'd like to bring up to begin with that I think should make everybody in the United States care about malaria, in fact, anybody in the world. One is to remind everybody that really malaria has only recently been eradicated from this country. Maybe most of the students in your classrooms would not have been born there, but their parents surely were alive during that, certainly their grandparents definitely. For example, this letter here, or postcard, is from 1962, and it's advertising malaria eradication. So up until the early 60s, late 50s, early 60s, there were still some homegrown malaria cases occurring in the United States. Something that people don't realize is that malaria is not right now, obviously, we think about it as a disease that happens in hot climates in the world, and there's no way in a place like Philadelphia that goes below freezing, especially after last winter, that we won't have malaria here. But in fact, malaria was rampant in the Philadelphia region. It was present in places as far north as Minnesota, and in fact, it was present, can go all the way up to Saskatchewan, Canada in the summers when it was present in this country. The mosquitoes, so malaria is a parasite that is carried by mosquitoes, and the mosquitoes actually still exist in this country. So the same species still exists. And really the only thing that has worked in us successfully eradicating malaria from this country is really a strong social infrastructure, and obviously the ability of a local and centralized government to actually control and implement disease control mechanisms. Obviously poverty has a big role to play with that as well, and we will see that fairly soon. So why should we care? So malaria is not in this country. Well, we should care really because almost half to a third of the world's population at any moment in time is a potential risk of contracting malaria.
Sorry.
Okay, there's no problem. There's
1 second. Boss, can I call you?
Continue.
Yeah.
You could pause.
Okay.
Sorry.
You can edit.
Feels like I'm in a production.
Sorry, boss.
No problem. No problem.
Very sorry.
We paused, so. Okay, shall we carry on?
Turn it off, yes.
Okay.
Sure.
All right. So malaria is, at any moment in time, about a third to a half of the world's population is at risk to being infected with malaria. So that's a big, significant portion of the world. There are actually almost 500 million clinical cases, 500 million clinical cases annually in the world, and up to 1.5 million deaths each year. Most of them children. Many of them, close to 80% below the age of 5. So that's very, very dramatic and very traumatic in certain parts of the world, and something we should care about not only because it's affecting people, but because we're human beings and we care about each other's safety and hopefully happiness. And obviously having a disease like this that's devastating in any country can lead to other things. It leads to the breakdown of social infrastructure, it leads to increased poverty, people cannot work, and obviously this decreases the stability of particular regions. So if we care, even if you want to remove our humanistic approach, you can think about just the world, the United States' interest in safety in the world and stability. And diseases like malaria are a leading cause of instability in the world. And so we need to really pay attention to malaria and work towards combating it. So this slide only shows you that the parts of the world, so the up here is all gray or white, essentially showing you that the northern hemisphere, obviously North America, all of Europe, Russia, and so forth, and also this, all the way down in Oceania and so forth, there is a very low risk for malaria. However, parts of the world, like in South America and as well as in Africa, there's a huge risk of contracting malaria. But these maps are actually quite deceiving. And so, for example, this map right here shows you which parts of the world are where malaria is distributed. And if you just look at this map, it seems like, oh, well, that's not a big deal. I mean, look, most of the world is gray. There's nothing there. There's no malaria. And so what I really like to show people, and this could be a really nice talking point with high school students, is redrawing these maps based on certain demographics and quantifications. So, for example, you can take a map of the world and redraw it like this. Obviously, I didn't do this. This is from Dorling et al., and it's all available online. But what you can do is you can redraw a map of the world based on where populations are. And you can see all of a sudden that India and China have expanded dramatically. Half of the world population lives there, basically. And then you have Africa, and you notice that the northern hemisphere actually has shrunk a lot. You can redraw the map of the world based on how much money is spent on healthcare. And so, for example, you'll immediately see here that Africa and South America, India, Southeast Asia, and so forth have pretty much disappeared from the map, whereas North America and Europe are gigantic. Russia looks kind of small, but there are not that many people there anyway to begin with, and compared to Europe and North America. But ultimately, what this tells you is that we spend a huge amount of money on healthcare here. We can afford to do that, whereas in many countries of the world, and coincidentally where malaria is, you don't have the spending of that amount of money, or the ability to spend that amount of money on malaria or diseases. And in fact, if you redraw the map of the world showing where malaria is, you will see that all of a sudden Africa and as well as parts of India have grown. In particular, Africa. Africa looks like it's the largest continent in the world. So if you're an alien coming to Earth and this is how you drew maps, you would think that Africa is the largest continent. And from the perspective of getting disease and getting malaria, it is. And so this is very, very critical and important. And it is actually, in fact, one of the leading causes of death and disease around the world. It ranks right up there with tuberculosis, which tells you something about malaria. Another very important factor about malaria, and in fact any of these diseases, is that they have become very adept at becoming resistant to the various drugs that we develop. And that's a very important and critical thing to think about. So we think that we spend billions of dollars to create a new drug, and that's it, that's going to solve it, that's going to be the cure for everything. And what it turns out that these parasites and bacteria and viruses, all of them, because of coevolution, they've been adapting with humans and other animals over the millennia. They're actually really good at adapting and essentially learning how to circumvent particular drugs. And in fact, this map simply shows you various drugs that malaria has become resistant to. Chloroquine, which was essentially was viewed as essentially that was going to end malaria, period. And in fact, for a period in the 60s, they thought this was going to be the case. Malaria cases dropped down dramatically. However, very rapidly, resistance to chloroquine came up. And all of a sudden, now chloroquine is not the drug of choice anymore. Most parasites out there are resistant. Other drugs are the sulfur drugs. Malaria has become resistant to them as well. Mefloquine is another one where drug resistance has appeared in particular in Southeast Asia. And finally, artemisins, which are basically the drug of choice right now. There's just recent reports that show that actually there is now drug resistance to artemisinin, and it's expanding. And very alarmingly, in the last few months, there was a report that of possible some artemisinin resistance in Kenya. And so that's obviously very scary. But historically, we know from the history of this parasite that this is actually we shouldn't be surprised, really. So I always like to also show a big tree of life. So we're looking here at a phylogenetic tree. And so again, this is a nice talking point with students to talk about phylogeny and what does it mean, how do you draw a tree. And in my handout, I have multiple links to different resources that talk about phylogenetic trees. And so this would be very nice for even the students to go in and build their own phylogeny for their family and so forth and learn about relationships. In the case of essentially a tree of life, so this is showing as much of life as possible on this tree. All of these little lines and connectors, which don't show very well on the slide, but they are essentially showing how different organisms are connected to each other. So you can see plants up here in the upper right-hand quadrant, animals up here on the left-hand quadrant, fungi are all the way down here. You have protists. So malaria is a protist, it's a protozoan parasite, so it's down here somewhere. And then you have bacteria and archaea. This is just a really small snippet. These actually would expand much larger if you open this tree up. And in fact, humans are all the way up here, and malaria is right down here. One thing that's really important to point out here is that the bulk of diversity on Earth doesn't come from animals. Doesn't come from multicellular organisms, which we are multicellular organisms. We tend to, because we are humans and we're very successful at living on this planet, and whether creating stuff or destroying stuff, we're really good at it. But we're actually the minority. And the vast majority of diversity comes from the rest of this tree, including billions of protozoa, bacteria, and fungi. So that's something very important to keep in mind. And all of these organisms have been essentially co-evolving on Earth for millions of years. And so what we see right now is obviously in the process of evolution, but has also been exposed to this evolution for millions of years, right? And so it's not something that's happening overnight. It's been going on for a very long time. These parasites have really adapted to us. We've adapted to them. And so, in fact, we talk about parasites becoming really better at infecting humans. But there are many human diseases or mutations that have occurred in response to parasites. For example, sickle cell anemia, which is common in Africa, is essentially natural selection for survivability, basically. So people who have sickle cell anemia, even though it's a debilitating and painful disease, they're more likely to survive a malaria infection than people with none. And so you can imagine how sickle cell anemia would have been enriched for and selected for in an environment where malaria is rampant. And there are many other diseases like this, and not only applying to malaria, but many other parasitic and bacterial and viral diseases. Okay, so malaria. So malaria is caused by an infection of a parasite. A parasite is a eukaryotic cell, just like our cells are eukaryotic cells. It has a nucleus, right? So that's one of the hallmarks of a eukaryotic cell. These parasites, they belong to a large family of other parasites called epicomplexan parasites. Okay, and these are thousands, over 5,000 parasites, maybe many more, and most of them are pathogenic. And so we know Plasmodium, it causes the disease called malaria, and there are many species of Plasmodium. The ones that cause disease in humans, there are 5 of them, but there are many other species that cause disease in, you name it, any animal you can find out there, there'll be a Plasmodium species that infects. There birds get infected with malaria, crocodiles get malaria, monkeys obviously get malaria, and humans get malaria. Related parasites are, for example, Toxoplasma gondii, which causes a disease in humans. About 30% of the US's population is infected with Toxoplasma. It's a cousin of malaria, basically. 80% of people in France are infected by Toxoplasma. And that's because they tend to eat more raw meat over there. And so it's transmitted by eating either fecal oral contamination or by eating raw meat. And cats carry toxoplasma. And that's a relative of malaria, and the many other ones. If you ever go to swimming pools, public pools in the US, there are often outbreaks of cryptosporidium, cryptosporidiosis, right? And cryptosporidium is a cousin of toxoplasma and malaria. So these types of parasites, the apicomplexan parasites, are everywhere. They exist everywhere, and they're many of them are disease causing. So malaria goes through this life cycle, which can be divided into different parts. Part of the life cycle takes place in the mosquito, and that's a sexual life cycle. Okay, so like any organism, or let's say most organisms on Earth have to mate. You need to exchange genetic material. Malaria is not immune to that, and they do that in the mosquito. Okay, and so this stage of the parasite is absolutely essential. Mosquitoes, in particular female mosquitoes, are the ones that are infected by malaria. And there's a reason for that. Talking about coevolution, so female mosquitoes, they're the ones who carry the eggs, and once they carry the eggs, they become very hungry, and they need to feed on blood. Males don't bite you. So when you get a mosquito bite, that's because a female mosquito bit you. And so malaria has evolved to actually grow in female mosquitoes, not male mosquitoes. Once the parasite goes through a developmental cycle inside of the mosquito, and all of these, when you read the names of these different stages, they sound really fancy and complicated, but it's just a development. Just like anything develops, you can imagine just the human embryo developing and so forth. There are different stages of the human embryo development. And the same thing with these parasites, they just give them fancy names. They have different names. You know, you'll hear things like sporozoids and merizoids and trophozoids and so forth. You know, don't worry about the names. The point is that these are different stages of the life cycle that are characterized by certain features, phenotypes, and locations where they exist. And so in the mosquito, you have the mosquito ultimately will bite a human and inject these sporozoids into the human. The sporozoids will rapidly go to the liver. That's the first place they go to. In the liver, they differentiate and replicate in the liver for a couple of weeks, and then they break out and infect red blood cells. And then now you have infected red blood cells in your bloodstream, and there it goes through multiple rounds of growth, replication, and bursting red blood cells. And I'll show you some videos and stuff in a bit more detail in a second. And ultimately, while these parasites develop, they can actually home and get to the brain, where you can get cerebral malaria. This is the actual part of the disease that causes death in most people. But also there's another stage called gametocytes, which is the beginning of the sexual cycle of this parasite that needs to be picked up by the next mosquito so the cycle can be continued. So if you were a scientist and you're trying to develop drugs or ways to combat this disease, you can imagine many different places in the cycle where if you interrupt it, if you can stop it, you can stop the disease. And if you can stop mosquitoes from being infected, you've stopped the disease. If you can stop the sporozoid from getting to the liver, you've stopped the disease. If you stop the red blood cell stage from growing, you stop the disease. If you stop gametocytes from forming, the ones that get picked up by a mosquito, you've stopped the disease. So these are many stages where the disease can be stopped. And so in a little bit more detail, with videos, so a mosquito bites you, and this is a video showing you you see these little bright dots going through. These are sporozoids actually in this, being injected basically into the skin of a volunteer in this case. Once it's injected, these sporozoids, this is now a view of the skin. So the sporozoids start to migrate and they rapidly move under the skin. And here's another one showing that in a getting into a blood vessel. Okay, and so now you see how it went into the blood vessel right here. So basically, there's a lot of activity going on right under the skin after the sporozoids are injected. Once they inject it, then the sporozoids make it to the liver, and so these are sporozoids sitting in the liver. They're extracellular at this stage, but then they will invade and infect a liver cell. And so this is a sporozoid that's going in and infecting a liver cell. They in fact go through multiple rounds of infecting one liver cell after another, and finally they settle on one, where you'll see this one now is coming out again. And so they do this in the liver actually, and then they ultimately then will develop in the liver into merizoids. Once they do that, these merizoids will break out. This is a merizoid, or a schematic of a merizoid. So it's breaking out, and it's going to infect a red blood cell. These red disc-shaped cells are red blood cells. and as you can see, the parasite will invade and go in. And the amazing thing about malaria or Plasmodium, what it does, once it infects a red blood cell, it secretes hundreds of proteins into the red blood cell and really redesigns that red blood cell. Completely redesigns it. It sends hundreds of proteins in there. And remember, a red blood cell does not have a nucleus. Okay, and so that's an important hallmark here. So that the cell, the red blood cell, has made all the proteins it's going to make in its pre-erythrocytic stages, in its early stages, and then now it's now a cell without a nucleus. Okay, and then inside of the red blood cell, you can see the parasite it moves around a lot. It's doing different shapes, and it starts going through the red blood cell stage of the parasite. Okay, so going from top to bottom, so in the red blood cell stage, they go through something called rings, and so that's the first 24 hours in the red blood cell. After that, they go through a stage called trophozoids, where they start making all their enzymes and different things. They start digesting the hemoglobin in red blood cells. Remember, red blood cells are essential for carrying oxygen to the different parts of your body, and they do that with something called hemoglobin. And Plasmodium has evolved to actually digest hemoglobin. And they digest it, and they leave behind what's this iron, essentially junk left over from digesting hemoglobin. And you can actually see that. If you look at infected red blood cells under a light microscope, you'll see this really bright, crystallized thing inside of these infected cells. And that's something that they leave behind, and then they burst out to reinfect new cells. Okay, and as I mentioned, the parasites really engineer the red blood cell, and this is an infected red blood cell. They have things called knobs, and they look like knobs on the surface of the red blood cell, and that contains a lot of different kinds of proteins that the parasite secretes and sends to the red blood cell. So essentially redesigns the red blood cell. So, and if you it turns out that these knobs are important for the ability of the parasite to attach and adhere to endothelial into blood vessels, basically. And so they attach to something called CD36, which is a receptor. And if they're knob positive, you'll see all these red blood cells that are infected, they attach to the surface. This is just an experiment with a surface labeled with or containing CD36. And knob negative surfaces red blood cells do not attach to it. So you can make parasites that are knob negative and ones that are knob positive, and the knob positive ones will attach, the knob negative ones will not attach. So that shows you that this is very important for the ability of the parasites to cluster and get stuck in vessels, and that's actually what ends up causing cerebral malaria. You have all these blockages of vessels in your brain, and you can see that in the cross section of your brain. You'll see all these vessels that are blocked with essentially where the malaria-infected cells just clustered and clogged them. Okay, and so finally you have this formation of these gametocytes. One thing that's really interesting is that for the longest time people didn't know where these gametocytes are forming. Are they forming in the blood? It turns out actually recent evidence is showing that that's probably forming in the bone marrow. And the parasites are actually taking advantage of essentially the normal formation of red blood cells in bone marrow, and they're actually growing in that sort of red blood cell, red cell differentiation center, basically in bone marrow. And so that's actually a very interesting and new discovery, which will probably lead to some new drugs. So we have female and male gametocytes, and those ultimately will then be picked up by a mosquito, and then the mosquito life cycle goes on, and you have reinfection of humans. Okay, so now we're going to switch gears a little bit to shopping. So we're used to shopping, at least we used to go shopping in markets like this, where you can go and you peruse through different items that you're interested in buying. You may pick something out, you try it on, you turn it back, and this process will take you hours, obviously, and then you have to go out of the house. Obviously now, most of us are used to doing something like this, right? We can go online, we can order whatever we want. And in fact, depending on the site you go to, and we're used to this on a daily basis, so you don't have to be a scientist to be doing this, right? We go to Amazon on a daily basis, and we configure things. We say, oh, I want to find blue t-shirts, or I'm looking for a power adapter for my computer. It's a Mac, and it's this model, and it needs this adapter, and so forth. If you go and try to book a flight, right? You can tell it where you want to go, But then you can configure. There are all these, and this is just Kayak, right? So you can go there, and there are all these little things that you can configure, including, what kind of a plane you want to fly on, how long the layover should be, whether the plane has Wi-Fi, and so forth. So there are many things that you configure. So we're used to doing that on a daily basis. Okay, so the Eukaryotic Pathogen Databases, which I work for, we're essentially trying to do the same thing for scientists, and to allow you to come in and ask questions and design experiments and configure things on a website based on underlying data. And the underlying data can include the genome sequences, it can include the annotation on those genome sequences, it includes all the data that's out there. So everybody's creating data, and because the computer power has become so much better, we're able to generate so much more data, we're able to store it, we're able to process it, right? So some of the data that we process today, 10 years ago would have been unimaginable. People would think you're crazy if you said, oh, we're going to be talking about petabytes of data. This was out of this world. And 10 years from now, people will look back at what I just said and think, I'm crazy that I that I didn't think that this is possible, right? And so the Eukaryotic Pathogen Databases includes many different websites which cater to different pathogenic organisms. And so, for example, there's one for amoeba. So if you're interested in entamoeba or acanthamoeba, these are different amoeba that grow out there, there's a database for them. If you're interested in cryptosporidium, there's one dedicated for cryptosporidium. If you're interested in fungi, there's one for fungi, and so forth. And Plasmodium is just one of them. And as I mentioned, all of these resources, the goal is that you can go to them and you can start asking questions about the underlying data. And so I'm going to run through one of these such questions. And then, depending on time, I can run through a live demo of the exercise that we had, or just opening up for discussion. So there are different portions of the website that are that you can configure. You can open up and view the various searches and decide what kinds of results you want to return. And so in this case, what I'm going to do is I'm going to run you through a series of exercises or questions where we're going to try and design a potential vaccine candidate for malaria. And you may be thinking, oh, who am I to be designing a vaccine candidate? But we can just think very simply and logically. Well, what is a good feature of a vaccine? What are we looking for, right? And typically a vaccine is something that's going to be, your immune system needs to see it, right? So in order for your immune system to see it, it needs to be exposed to the immune system, right? And it needs to be coming from the parasite, right? And so there are many types of questions you can ask. You can ask to return for types of proteins from the parasite that may give you a short list of proteins that may be potential vaccine candidates. And I'll go through those questions right now. So one question you may ask is, well, cellular location. Where is that protein? Where are the proteins located? You may say, well, I want to have all proteins that are secreted. Okay? And you can do this by something called signal peptide. And so proteins that are, and you may have taken this already in your biology classes, but proteins that get secreted, they have something called a signal peptide on them, or secretory signal peptide. And that gets processed when they get translated into the endoplasmic reticulum. The signal peptide gets processed, and then these proteins in the endoplasmic reticulum get secreted out into different portions of the cell. Some go to internal vacuoles or organelles. Others go up to the surface of the membrane, and others even go out of the cell. Okay, and we can predict that signal peptide. There are algorithms out there that will predict that. We've done that, and so you can go in and say, give me all genes in Plasmodium that have a predicted secretory signal peptide. And so once you do that, you select this option. You can select which organisms you're interested in. You'll notice here there are many different organisms. It's not only Plasmodium falciparum. So Plasmodium falciparum is the form of malaria, the species of malaria, that causes the most death in the world. But then there are other ones. Vivax also causes disease in humans. Nulzi can cause disease in humans. And then the other ones, Burgii is a rodent malaria. Uwelii or Shabauti is a primate malaria, and so forth. And so they infect different species. But they're really useful for cross-species comparison and learning things about evolution. So once you run this search, you get a little button up here that shows you I have over 6,000 genes that had a predicted signal peptide in all of these species of malaria that are in the database. Okay? So now we have 6,000 or 6,500 results. And you may decide, okay, I'm done. I have my vaccine candidates, right? So that's an option, but that's still a huge amount of genes. If you were in the lab and you're trying to work on these, it would be very hard to work on 6,500 genes at the same time, right? So you want to try and narrow this down a little bit more, right? And so you can add to this and say, well, things that have a transmembrane domain, right? So proteins that have a transmembrane domain that makes them integral membrane proteins are ones that probably also go to the surface of the cell. So how about if I combine and add a step? So you can do this. You can click on Add Step. I'll actually show you a couple other analyses, so you can quickly, if you want, make a word cloud of all the names of the genes in here, so you can quickly see what's in there and see if anything sticks out. But anyway, you can add a step, and you can grow this strategy. So we're essentially doing what we call an in silico experiment. We're doing an experiment here that's online, right? We're asking, we already did an experiment. We said, give me all genes that have a particular single peptide, right? This is already a result of an experiment. It's not your traditional experiment where you've done something in a test tube, but nevertheless, it's a result. Now we're going to add a step to this experiment. We're going to grow it, and we're going to say, well, how many of these single peptide containing genes also have a transmembrane domain? And so we can add transmembrane domain searches. We can configure the search of the transmembrane domain searches, decide how many transmembrane domains we want, and decide how we're going to join the results. So you can intersect the results. That means you want things that have a single peptide and a transmembrane domain. That's fairly restrictive. Or you can use a union, for example, and there are other options too, but a union would allow you to say, well, give me things that have a single peptide, things that have a transmembrane domain, and things that have both. And so in this case, that's what I selected. And so now all of a sudden you have over 11,000 genes, right, that have either a single peptide, a transmembrane domain, or both, right? And that makes sense. You have all the ones that are transmembrane domains, all the ones that have single peptides, and all the ones that have both, okay? So now we have even more. But now we've cast a really wide net. So now we need to try and pull this back a little bit and see if we can narrow it down. So another question you can ask is something about polymorphism. And so if you haven't talked about polymorphism in your classrooms, this is a nice point to stop and talk about genetic diversity. Right? So if you look at my genome and you look at Kristen's genome, right, we're probably going to be mostly identical, right? But we don't look identical. We're not twins, right? So you will find some polymorphisms that differentiate us as well, right? But in general, we're fairly identical. We biologically we function the same way, and so parasites are the same thing. If you go out there and sequence genomes of hundreds of parasites and you compare them at the sequence level, you'll find that there's a lot of similarities between them. But then you'll find that there are mutations, and if you can correspond these mutations with a phenotype. So if I can say, oh yeah, the mutation that Kristen has in this particular gene gives her, dark hair, or she keeps her hair and I lose my hair, right? You've already now linked a mutation to a particular phenotype, right? And so same thing with parasites. You'd like to be able to compare many isolates or strains of the parasites out there in the field and find mutations that tell you something about the underlying genes, right? And there are different kinds of mutations, right? You can have a mutation that is synonymous, which means, and I'm sure you guys have talked about how a mutation in a DNA could cause a change in the amino acid sequence, right? And so a synonymous mutation is one that does not cause a change in the amino acid sequence. A non-synonymous one is one that causes a change in the amino acid sequence. And so those are the ones that would potentially change the function of the protein or change the way it looks to the immune system. So remember, we're trying to design a vaccine candidate. And if I were a parasite, right, and I'm sending out these proteins to the surface of the cell, and the immune system is seeing them, I'm dead, right? The immune system will kill me, right? So if I were a parasite, I'm sending these proteins out, I would really want to make sure that the proteins are changing, right? So that's one way of looking at it. So that will be a selective pressure on you to make sure those proteins that go to the surface have variability, okay? Just like our antibodies have a lot of variability, and our bodies have evolved, our immune system has evolved to generate a lot of variability so we can recognize any kind of foreign protein that comes into our body, right? Parasites and organisms have also developed their own ways of variability so they can actually circumvent the immune system, right? And so malaria is not immune, they've done that as well. And so we can, without going into the details in too much, but we can go in and compare a lab strain with a field isolate, and we can say, I want to find all genes that have non-synonymous mutations, and I want them to contain at least 4. I just picked 4 as a sort of a higher than average number of non-synonymous mutations. So you can increase that or decrease it as much as you want. And I'm going to intersect my results with this result right here. Remember, we're growing this search strategy. So once you do that, now we're down to 396 genes. We went from 11,000 genes to only 390, or some about, let's say about 400 genes that are potentially secreted have at least 4 non-synonymous mutations. So now all of a sudden we have a much more manageable number, right? And sort of using the same logic, we can grow this even further. And so I'll skip over this one here. We can say, if you had if you were trying to develop a vaccine, you're working in a company or in a lab, and you're trying to develop a vaccine, one thing that's really important, you want to make sure that this vaccine is not recognized as a self-antibody antigen, right? Mm-hmm. So you don't want to make something that then your immune system goes crazy and starts attacking it, but then it also recognizes something that's normally found in your cells, because then you'll have an autoimmune disease, right? And so you want to make sure that that any of these genes that are found in your experiment do not have homologs in humans, for example. And so you can do this. So there's a particular search that you can run called a phylogenetic pathogen search in Plasmodium. And so you can go in and say, I want to find things that are conserved in, not conserved in mammals, not conserved in arthropods, for example, the insects, but present in the parasite. And now you have, we're up to, I did another step here where I converted this to the orthologs of all Plasmodium species. So now we're up to about 2,600 across all Plasmodium. But if you look at falciparum, down here, there are about 648 genes that actually met all of that criteria so far in my searches. Okay, and then I'm just running through a bunch of these, so you can also look at gene expression of these genes, when are they expressed, and so forth. But you can make these searches as long or as short as you want, but ultimately you have a list of results. And people tend to, if you went to Kayak and you booked a flight, and then you just blindly purchased a ticket, right? You didn't check your work, right? You might be flying somewhere you don't want to fly, right? Or you might be going first class instead of this or that, but you're paying all the money, right? So anytime you do this, you check your order. You actually look at the results of your search. You say, oh, is this flight really going to Phoenix where I wanted to go? Am I paying the right amount for it? Do I have the right seat, and so forth? And it's the same thing with any kind of data in a scientific database. You as somebody who's studying science, you should be able to look at this and say, well, do the results make sense? Am I getting things that are useful, that actually make sense, that it would be in this category? And so, for example, as we look at the list of results, you'll see there are things called ring-infected surface antigen. These rope tree-associated antigens. We can keep going down. You have a number of conserved ones, but the ones I showed you before are actually known proteins that are being worked on as potential vaccine candidates. So those are your positive controls in your list of results, which is really good. And the fact that you have many things that are just, they don't know what they are. They're there. They know these genes are there. That tells you that there's a nice sort of list of genes that you can start working on and looking at. Any one of these could be the next important vaccine that will be discovered for fighting malaria. So one nice thing about Plasmodium, which is the database that is in the exercise that you can use in your classroom, is that you can create a login account which will save all of your searches, and you can share your searches. So as an instructor, you can have your students go in, do a homework assignment or a lab assignment where they run such an experiment, and then they share with you their results. And then you as an instructor, you can later on that evening, you can click on their links that they sent you, and you can see all of the results that they ran, and you can actually see if that made sense and come back and discuss it with the students and see how you can improve their searches and their search strategies. And so that's a very powerful tool. Here's an example of a workshop I ran in Singapore last year, where we do this now regularly in all of our workshops. But here's an example of the output. So the 3 different groups of students. And again, you can do this as group projects, which is really useful. And each one of them came with a different strategy to identify a potential vaccine candidate. And any of these is good and a possibility. All right, and then the last thing I want to cover is this notion that we've been running these searches where you come back with lists of genes, but you could also ultimately just go and visit one of these genes, the gene pages. And it's almost like looking at a Wikipedia page of the gene. Mm-hmm. And those gene pages are just full of information. And any of these gene pages can be used as a sort of a backdrop of an entire sort of biology class lecture, because you can talk about genomes, you can talk about chromosomes. In the case of parasites, you can, like Plasmodium, they have a haploid genome. They're not diploid like us, and so you can talk about that. You can talk about gene structure. So these are we're looking at a particular portion of chromosome 11 in this case, and here's a gene we're looking at, but there are genes upstream and downstream of that gene. They have different structures. Some of them have introns in them. So immediately right here, you have a whole, you can talk for at least 15 minutes about just gene structure and so forth as a backdrop to talk about genes in general. You can talk about polymorphisms, which are there as well, and start talking about population diversity. We can go down and look at the protein section, where you can see, for example, here's information about a signal peptide. There's information about different domains. So you can talk about how a gene makes a protein, and a protein forms structure, which has particular domains and interesting parts that would be that allow you to define the function of the protein. And so that's an important thing to talk about. There's a section about expression, so gene expression. So now all of a sudden you can start talking about, RNA. And what does that mean? And in the case of Plasmodium, it does something really interesting where it has this cyclical expression pattern in the red blood cell stage. It starts up, it goes through this sort of low expression and high expression. So many genes have this cyclical pattern of expression, which you can then talk about how are genes regulated, what dictates that, and so forth. Many different data types. There's some data from actual patients. Let's move on here. You can look at actually a list of results and look at them actually on a genomic context. So this is all the chromosomes in malaria and the location of all the genes in our results. So there are different sort of visualization tools that allow you to do this. And that's pretty much it in terms of the lecture portion that I want to talk to you about. And ultimately, the tools are there. It's all free. These are all free resources.
Plasmodium, right?
This is all Plasmodium, yes. But all these tools and all these things can be run in any of the resources that we have. The same types of exact searches and so forth.
Okay,
Yeah. And the exercise that I have prepared for today is an exercise that takes you to Plasmodium and asks a very simple question, which is, can you find any gene, all genes in Plasmodium falciparum that have at least 10 transmembrane domains, right? And so looking at the website, you can go down and sort of look at this. Here are all the searches that return genes, and you can start seeing, oh, these are all different categories. It's like a text search. You can go down to genomic position. But transmembrane domains are really protein features or tell you something about cellular location, right? So so we have it actually in both places because different people think about this in different ways. You can ultimately find your transmembrane domain search, and now you can really configure the search as you wish. So I can say I want to clear all of these. I only want to get Plasmodium falciparum 3D7 parasites, and I want to get genes that have at least 10 transmembrane domains, and a maximum of 10, because that's all I want to get. I want to get any gene in Plasmodium falciparum that has 10 transmembrane domains, right? And so when you run this search, you get 27 genes in your result set, okay? And we can go down and look at these, obviously. And so these are all the genes that have had transmembrane domains. And the reason why I ran this search is for a particular reason, because I wanted you to get to this gene called chloroquine resistance transporter. And so remember I mentioned earlier in the talk that chloroquine used to be thought of as the holy grail of to get rid of malaria, and it turned out the parasites became resistant, and it's because of this gene right here, this parasite gene. So the parasites that had mutations in this gene, which allowed them to essentially pump out chloroquine from the cell, were able to survive, and they were selected for, basically. And so you can go to any of these genes by simply clicking on the gene ID. And when you go there, you get this sort of Wikipedia or encyclopedic version of or view of a gene. And you can go down and look at exactly what I was showing you in the presentation. But here's the gene. It has multiple exons. So these are not the transmembrane domains, but these are the exons of the gene. So it's a multi-exon gene. And you can just scroll down. And what you would be interested in doing is going down to the protein section and seeing where these transmembrane domains are. And here are the transmembrane domains, and you can count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? And you can talk to your students about what are transmembrane domains, what are the features of the transmembrane domains. And then you can go down and look at the actual sequence, the amino acid sequence, and see if that makes sense. Can you find stretches of hydrophobic amino acids in there? Let's say about 25 or so, that are, so can you find 10 of these clusters, and so forth. And so, again, it seems like a very, very simple exercise, but it's really meant so that you can actually then use it as talking points for various different parts.
Protein structure, then the DNA sequence, and the protein sequence,
right.
and the comparison.
Exactly.
And also gene expression, obviously.
Yeah. So, and that's all I had for today.
That's nice. That's very nice. And my question will be that all these high school students, number of cases, they find out this is really intimidating to them.
Right.
Yes.
Not only, let me tell you, it's not only high school students, even scientists find this intimidating.
Yeah.
You know, people who have PhDs.
Yeah. So they find out it's quite intimidating. And normally I start with 2 comparison with the +10 and then gradually start adding more and more material, and then I normally ultimately go up to 7, 8, sometimes 10, depends on the student body.
Right, right.
And then ask them to rank it, and then after that I ask them to go for the protein sequence +B, and then again rank it and see that, ask them that whether these 2 ranking match.
Right. Yeah, you have to really, you know, design sort of exercises that are fairly straightforward, that they don't get sort of bogged down by some complex techniques and so forth. And so,
no, if I go anything beyond this, means they go crazy.
yeah, it gets very hard. That's understandable. Yeah. But I think it's also important to show them that these tools are available. So for the students who are really, wanting to explore more, those things are there, and it's really not. There's nothing magic about it, really.
That's the point. Means that to them and work with them. That this is not magic.
Right.
Yeah, this is definitely not magic. For most of the students, they think that, well, you're telling us all this kind of stuff. How do we know this?
Right.
And then gradually fit them into this.
Right. Yeah, and I mean, our topic, and I'm sure you can appreciate that, but a topic like malaria, because there's a lot of genomics data about it, and there's a lot known about the parasite and the disease it causes. I mean, that alone can be a topic for an entire, semester of AP biology, right? Where you discuss everything from disease to just basic genetics and gene structure and so
AP
forth.
biology is now, they restructured, the College Board restructured the AP biology last 2012. They completely revamped AP biology. And now it is more towards the cell biology.
Right.
Yes.
Yeah.
So this malaria actually helps.
Good. Yeah,
Because
absolutely.
It is a eukaryotic system rather than always going back to the bacterial
That's right.
system.
That's right.
Yes.
And there's a lot of good cell biology in
Yes.
Plasmodium as well. Yeah.
Yes.
Yeah.
So that, and also the infectious organism, and also how means the human body is also eukaryotes. Malaria is eukaryote, Plasmodium is eukaryote,
Right.
and how these 2 interact.
Yep. Absolutely. Yeah.
That actually helps a lot. Any means it seems to me you get this lecture to a lot of places, means anywhere the undergraduate students, any kind of interesting questions?
Yeah, I mean, yeah, absolutely. There are always interesting questions, and again, it's a variety of levels, obviously. So most of the students I work with are graduate level students. They're in graduate school, and so next week I'll be traveling to University of Georgia and I'll be running a workshop there. But it's not for students from the University of Georgia. It's students from all over the world actually who'll be attending this workshop, and they're mostly graduate students. And they're always very interesting questions. But I do have some workshops that end up including a number of undergraduates as well. And definitely there are some always interesting questions.
Any specific type of question?
I wouldn't categorize them as types of questions. Mostly, people are very obviously curious and inquisitive about how to actually use the database to its fullest potential. We end up doing a lot of sort of covering the underlying techniques. So for example, if we talk about microarrays, which are used for looking at expression, or RNA sequence, looking for expression, then we obviously have to sit back and say, well, how is RNA sequence data analyzed? How do you map it back to a genome and so forth? So it ends up being a lot of sort of talking points trying to explain the technology behind a particular result. A lot of times also, obviously with when you get a database that has a lot of data that's coming from primary research out there, it's the publications are there and available, right? And so depending on the level of the students, some of these publications are actually useful for even a high school level too. And then
once they understand this part, then they can read and get it.
Right.
Yes.
And imagine that if they read a paper, and then they can go to our database and actually ask questions about the data that's coming from that paper, as opposed to saying, oh, we see these beautiful figures, we'll just believe what they say. Right? This way they can actually go and say, well, let me change things. Let me look at this and that. Yeah. Exactly. Yeah. So.
Means ultimately they can read these papers. Means it's a little bit haltingly, but they can.
Yeah.
read this paper, at least get the abstract.
Right.
Get the
Well, one thing that's important is actually also teaching sort of how to read a paper, right? How to read a scientific paper. So it's not about how many papers you read or how complicated a paper is, but really how to read the paper. What are the things that you should be looking at? How do you understand the methods? How do you take that and look at the actual statistical methods they use in the results?
Yeah.
Do they make sense? Are the results good or bad? Do you agree with them, disagree with them, and so forth? Yeah.
That way, one of the things College Board did means it's a very nice thing in the second paper, the biology, first two questions, they have to, means the data is not given, it's a description.
Right.
They have to pull the data out, then they plot it.
Oh, I see.
Okay?
Yeah.
And once they plot it, they have to calculate it.
Right. Right.
Once they have the data, then the result.
Right.
Then they have to find out that whether this result actually supports the hypothesis
Right.
or it does not support the hypothesis.
Right. Right. Exactly.
Okay.
Yeah.
Okay. And then they will also, normally there is always a question with geographic variations, and then what
Right.
happens. Yeah.
Right.
What
Right.
happens with
Yeah.
geographic variation. That way they designed it very well. The big problem is that most of the students have really hard time
Yeah.
with
Yeah.
this kind of analysis.
It is difficult. It's not easy. It's not
Yeah.
easy. But it's and then it actually takes a lot of work to actually get them to get over that sort of
That hump.
block. Yeah, that hump
That-
exactly.
that
To
hump.
start
Yeah.
thinking that they can do it.
Yeah.
Yeah.
Means most of the cases, the biology students, they think that, okay, I'm in biology, I'll just memorize anything
Yeah.
that come in front of me.
That's right.
And then you basically just go and answer the question.
Right.
The analytical mind in the biology, that, okay, do I really have to do this?
Yeah. No, that's true. That's true. Yeah.
But that was wonderful.
Good. Good. Well,
That was
thank you.
wonderful.
Yeah. Thank you.
Okay, and.